Hi, welcome to my presentation on designing new inhibitors for an enzyme called lactate dehydrogenase. So let's talk a little bit about the background behind this project and why it's important to inhibit this enzyme in the first place. All right, first off, starting with a little bit of background. We've all hopefully talked about this a little bit in biology, metabolism, how our cells make energy. So our normal cells, as shown in the picture on the left, we take in glucose, which is basically sugar, the food that we eat, turn it into two molecules of a compound called pyruvate, and then we turn that pyruvate into energy for the cell. However, cancer cells are a little bit different. Instead of turning that pyruvate straight into energy for the cell, they take the extra pyruvate they have that's left over and turn it into a compound known as lactate. Why is this important? For a couple of reasons. First of all, imagine me and my sister, we're playing with toys in the game room, right? And if I act like a cancer cell, I can take up all the toys in the room and just put them into a box. I can turn those toys into lactate essentially and put them away in a box. And because I have no limit on this box, I could take as many toys as I wanted. Meanwhile, my sister doesn't have a chance to play with any toys because I keep on taking away all of them. The same thing is happening in our bodies. Cancer cells have the ability to take up as much energy as they want, stored away in the form of lactate. That's depriving nearby healthy cells of the energy they need to survive and grow and divide. Secondly, lactate has been shown to mediate tumor progression, which means it helps the tumor get worse. It helps the tumor metastasize, which means it helps it spread from one part of your body to the rest of your body. And usually this leads to very negative adverse effects for the patient, including fatigue and usually death. Our goal in this project is to find a way to cut off this pyruvate to lactate transition so that I can stop hogging toys, just like our cancer cells can stop hogging energy. The enzyme that's the key player behind this conversion of pyruvate to lactate is called lactate dehydrogenase. And technically, it's not one enzyme, it's like a family of six. It refers to a family of 6 different kinds of enzymes. The reason there are different kinds is because each enzyme, each lactate dehydrogenase, is made of 4 subunits, and each subunit is usually one of 3 types: A, B, or C. Usually it's only A and B that really play a role in cancer, so we'll focus on those in this project. The A subunit is usually found throughout our body and muscle cells, whereas the B subunit is usually found in our heart. And they differ in their binding affinity to the substrates and their ability to catalyze the reaction. So what are the substrates? Well, the leading theory is of an enzyme and its substrate is like a lock and key model. So imagine this lactate dehydrogenase like a lock. We need 2 keys to be able to open this lock. The first key is called NADH. NADH binds to lactate dehydrogenase, changing its shape, allowing for the second key, pyruvate, to enter. And once all these components have assembled, the reaction can proceed. Now, here's the issue. Each subunit has its own catalytic site, which means if you just took an A subunit or just a B subunit, it could carry out this transformation from pyruvate to lactate. But if we inhibit only one of the subunits, let's say we only inhibit the A subunit or the B subunit, then that won't actually stop the enzyme as a whole from functioning because it is composed of multiple different types of subunits. The issue is current preclinical inhibitors or lab inhibitors that are being developed in the lab have only been engineered to inhibit either the A or the B subunit. My goal is to create a new compound that can inhibit both the A and the B subunits at the same time. Why is this important? Because, as we'll see in just a little bit, it has many effects beyond just better potency and better cancer treatment. So another quick overview. First of all, there are no clinically approved LDH inhibitors. So if you went to your pharmacy and you asked for a lactate dehydrogenase inhibitor that treats cancer, you won't find any. And secondly, the ones that are being developed in the lab, like I mentioned, only target one of the subunits. My goal is to target both at the same time. And we can do this using a completely on-computer modeling based project done straight from home. Why is this important? First of all, we can limit the lactate energy storage mechanism. Basically, what that means is we can stop cancer cells from hogging all the toys in the body. Secondly, we can lower the side effects of lactate presence. So earlier we discussed how lactate leads to the spreading of the tumor to different parts of the body, the growth of the tumor. We can limit that by cutting off how much lactate is produced. And finally, if we have just one compound that inhibits both subunits, instead of having clinics to keep in stock two different compounds, one to inhibit the A subunit, one to inhibit the B subunit, we can keep it logistically simple and we can lower patient healthcare costs by just keeping one compound that's capable of inhibiting all the subunits and the enzyme as a whole. So with the importance and background out of the way, let's get into how we actually develop these inhibitors. So our approach was first to investigate current preclinical inhibitors, so drugs that are being developed in the lab. Second, to create models around those inhibitors and run new molecules through those models to see which new molecules are most likely to work. Thirdly, we more accurately modeled their efficiency against the enzyme by using a program known as molecular docking. And we'll talk a little bit more about that later in the presentation. Finally, we evaluated their safety and specificity, or at least we predicted it based off of online web tools that already exist. So let's take a look at the preclinical inhibitors. These are the 5 major ones we researched through a thorough literature review, and the 2 we ended up picking for further studies were GSK, which is a competitive inhibitor. In other words, when you have your enzyme, GSK will enter the active site shamelessly and it'll stop pyruvate from entering the enzyme. So that stops the reaction from happening. The other one is RGNE. RGNE is allosteric, meaning it binds to a different part of the enzyme, changing its shape, so now the enzyme can't bind to its substrate anymore. So for GSK, you could think of it like a mimic key that's blocking the keyhole, but for RGNE, you can think of it like a welder who's completely changed the key itself. It's completely changed the lock itself. So now the key that used to work won't work anymore. Now we move on to pharmacophore generation. So what is a pharmacophore? Well, what we used is molecular docking to put this inhibitor, GSK or RGNE, into the lactate dehydrogenase enzyme. So what molecular docking is, it takes the ligand, it evaluates different conformations through random simulations, and it sees which of those conformations is most strongly bound to the enzyme. And the idea is the more strongly bound it is to the enzyme, the more likely it is to be able to inhibit the enzyme. So the better this mimic key fits in the lock, the more likely it is to stop the real key from getting in. After molecularly docking these compounds to the enzyme, we can use software to evaluate the interactions between the enzyme and the drug. So this is an example of GSK, one of the inhibitors from the last slide, interacting with the enzyme. And you can see certain amino acids are having certain hydrogen bonds or van der Waals interactions, different types of interactions between the atoms of the ligand or the key and the amino acids of the lock of the enzyme. Based off of this, we can analyze these interactions and create a sort of mold around this compound. So imagine we come up with a cast that's a specific shape. When we create derivatives of these inhibitors or new modifications of these molecules, we can generate hundreds or even thousands of them and run them very quickly through this mold, through this cast, and see, okay, which ones are most likely to fit? The ones that are most likely to fit are the ones that are most likely to outperform or do at least as well as the preclinical inhibitors mentioned from the previous slide. So what we're looking for is, through this pharmacophore, we get a computationally efficient, which means a really fast and it doesn't take too much computing power. We can create a mold and we can run new molecules through that mold to see how likely they are to work. So we got 7 GSK derivatives and 12 RGNE derivatives that worked. After that, we took those compounds that passed the mold and we used molecular docking against randomly simulating to see how well the key fits in the lock. And we found 2 derivatives, G45 and R118, that fit the lock really, really well, and even statistically significantly better than their parent compounds, the inhibitors that are currently being developed in lab. So we took those further to evaluate their safety and their specificity. So just to quickly run over what this table is showing, for the G derivatives, so these first 2 rows over here, G45 and GSK, I want to focus on the Lipinski and the SA. Lipinski is the number of rule of 5 violations, and that's basically just a golden standard for what does a drug molecule look like. So our compound actually does better than GSK. It looks more like a drug compound. And secondly, its synthetic accessibility is lower, meaning that it's easier to make theoretically in the lab. Now, if we look at the R derivatives, R118 versus its parent RGNE, I want to focus on its specificity. So what this is telling us is the R118 is very specific to LDHA, and it can target LDHA. Of course, ideally, it would also be able to target LDHB, but computer predictions say that it's more likely to focus on A instead of B. But that's still better than what we have in the lab right now, RGNE, which actually targets neither of them. It's more likely to target other unwanted proteins in our body and have unwanted side effects. So both our derivatives G45 and R118 outperformed their parents in terms of some aspect of safety or specificity. So this is very promising. And again, the impacts of this project could be lowering healthcare costs for patients because they don't have to buy two different drugs, they only have to buy one. It can improve survivability for patients because we slow down the progression of the cancer tumor, we slow down the metastasis, and we can help patients spend more time with their families and spend less time in the hospital. And finally, an important implication of this project is it was completely done on the computer in silico. And this shows that all you need is a computer, enough ambition, and the will to find free resources online, and you can do any research project to develop an inhibitor against any enzyme, not just cancer enzymes. Finally, I'd like to give a huge thank you to my mentor Joshua for his invaluable feedback and his priceless skills in this field of cancer biology. He really supported me by recommending me tools and helping me do a lot of thorough Google searching. And finally, I want to thank you, audience, for listening to my presentation. I hope you learned a little bit more about cancer and how we can stop it from progressing and hurting patients. Thank you for your time.